Well, good morning, everybody. I wanted to talk to you today about functions of genetic information. I appreciate Dr. Wood's talk before this because it actually covered a lot of stuff that I don't have to talk about now because the ground has been covered. But I want to review a few concepts because I know not everybody in here necessarily has studied this in detail. So I want to review a few concepts and move forward. And with the main idea here being I want us to understand that there's a lot more going on in the genome than I think was initially thought. And I want to present to you a few of the things going on that I think are really interesting and open some doors to maybe areas of study and things that we can examine. So everybody's familiar probably with the general double helical structure of DNA. I got this model sometime last year and just thought it's a great way to teach my students. I use this from time to time with them just to get the concept of we've got a linear array of bases, right? We've got a sequence of DNA, which would be all the bases, but they're paired up across this double helix, and then that's twisted up in a cell. And I've spent most of my career studying the double helical structure and what that does in terms of what we call topology in a cell. In other words, a chromosome is much larger than this. I don't know, Dr. Wood, the average human chromosome is probably what, 80, 100 million base pairs, something on that order. So pretty long. You know, this has got maybe 20 or so nucleotides, right? Amplify this out, you know, by 100 million base pairs. That's a really, really long piece of DNA. Or, as I like to illustrate sometimes to think about it, that's a really big rope, right? If you just think of it even as a rope, this one easily got entangled sitting in a bag for a while. And so you can imagine there's a lot of problems that come from that. We'll talk about that a little bit later. But it's that information in DNA that our body will use to make RNA, which can then be used to make protein. And so it's this idea that has been termed the central dogma of molecular biology, which in essence is still true, but it's not the whole story, right? This is part of the story. And there's more to it than this. But this is generally the idea is that there is information encoded in DNA, which would be genes, as Dr. Wood was talking about. They can get transcribed to make RNA. Some of that RNA is messenger RNA that gets translated to make a protein. And so we might call all of the DNA, the chromosomes, the genes, our genome. All of the RNA might be the transcriptome. So all organisms have somehow, somewhere, a transcriptome, which would be all of the RNA molecules. And then all the proteins made would be the proteome. Now, it goes past this. We'll talk about that a little bit later. There's more to it. But we've got basic processes. We talk a lot about replication, which is the copying of DNA, so making a copy of it. Transcription being making RNA from the DNA, and then translation being making a protein out of that. And it comes down to this, and that is we've got this sequence of nucleotides which code for a particular sequence of RNA, which then get translated into individual amino acids in a chain. So every 3 nucleotides codes for some unit of that, right? So 3 nucleotides gives us 1 amino acid, and we chain all that together to make a protein. And so there's genetic codes. It's interesting, we talk about the genetic code. I actually looked it up the other day just to see. I don't remember the number, but there are a few dozen genetic codes actually, because not all organisms encode all amino acids in exactly the same way. So there's some variation. There's a lot of commonality, right? But it's not universal. So I think that's interesting. So as Dr. Wood talked, if there's only 2% of the genome codes for protein, what's the rest of it doing? That seems like a lot of wasted space. And that's been part of thought in the field that I've studied in for a long time, and that is, what is all the rest of that DNA doing? And some people have considered some parts of it to be junk, and some people have considered some parts of it to be vestigial or leftover. It just depends on who you're talking to and what context they're referring to it as. I like to think of the cell a lot like you might think of a computer system with a bunch of different components, right? So cells have organelles, and those organelles all have various functions, and you've got to have all those functions to make a cell that works, right? If you don't have mitochondria, you're not going to be making a lot of energy, right? You can still do glycolysis and get a little bit out of it, but you're not going to be getting very far, right? So the idea being, if you don't have some of these compartments, right, you're going to be missing out on things, right? Our red blood cells cannot replicate themselves. Why? Because they lose their nucleus, right? So they terminally differentiate, they become a red blood cell, they circulate in your bloodstream for 100 days, something like that, 120 days, and then they die. And that's it. So all of these different components are in some sense analogous to the components of a computer, right? You've got a hard drive, you've got a video processing unit, you've got RAM, right, which is your random access memory where you're actually accessing things actively. So you got all these different components, and I like to think of a cell a lot like that. And we're not the only people making these analogies. So here's a recent paper from PNAS, I say recent, it's from 2010. A relatively recent paper, where they said they're comparing genomes to computer operating systems in terms of the topology and evolution of the regulatory network. So I'm not a Linux programmer, some of you may be, and this may make a lot more sense to you, but they said, look, here's a Linux call graph, and here is an E. coli transcriptional regulatory network. And they, in the paper, compare and contrast, what do you see that's similar, what's different? So I want you to realize we're, as creationists and as folks who believe in design, we're not the only people asking these types of questions and making these types of comparisons. I think it's a valid comparison to think of that. And I actually like the things they came up with. And one of the things that I thought was very interesting in this table, if you look with me, so under origin of evolutionary changes, random mutation, natural selection for E. coli, look at what they put under the Linux call graph. Designers fine-tuning. So that was kind of an interesting comment for them to put it, but that's the analogy they made there. So I think they're more right than they know. So living systems are organized in layers. And I talked about this the other day in our biochemical basis lecture, and I'll talk about it some tomorrow as well. The idea that as humans, we've got organ systems, and our organ systems are made up of a series of organs, which are made up of tissues, which are made up of cells, which are made up of organelles, which are made up of biomolecules. And the four main biomolecules we think of are amino acids, which make up proteins, nucleic acids, which make up DNA and RNA, lipids, which make up phospholipids, cholesterol, etc. And then we also have carbohydrates, so glucose, but all of its other friends and neighbors and cousins, right? So you've got large sugar involvement in various ways inside and outside of cells. So to me, there's a lot of cool stuff going on there, and I think it's really neat to consider the layers of organization, and that's a lot like what we see going on with the system. So we've already talked about what a genome is and DNA, and you've seen the structure, and how do we actually study this? And this is something that I know Dr. Wood has specialized in for a number of years, but I just wanted us to take a look at a chromosome for just a minute. This is just using the freely available NCBI website, which allows you to look at various sections of DNA. And I've pulled up here one of my favorite chromosomes, chromosome 17, right? Everybody's got a favorite, right? Anybody else have a favorite chromosome? Okay. Everybody's got a favorite chromosome. This is chromosome 17, and what you're looking at there is the entire stretch of chromosomes, so 80 million base pairs. So you cannot even see the nucleotides, right? Because there's so many, you can't fit 80 million across one screen. So let's zoom in for a minute and take a look. So if we zoom in here, and this is zoomed all the way in, what we're looking at, if you can read it right here, it says 82 base pairs. All right, so we've zoomed way in from 80 million down to, we're just looking at 82 base pairs, and that's what these A's, T's, C's, and G's are across the screen. Okay, so just wrap your mind around that for a second. We've taken a whole chromosome, 80 million nucleotides, and we've zoomed in on 82 base pairs of that. And that's pretty interesting when you think about just the ability to do that, to zoom in and say, here's a particular sequence of DNA, right? These are nucleotides in our genome. And what you see at the bottom there is that green bar. What I want to show you is as we zoom out, that green bar is going to become something different. So if we take a step out, and now we're looking at about 1,500 nucleotides, that solid green bar that we were seeing is now a much smaller bar. And what we know is that's actually an exon of a protein. So proteins have exons and introns in their genes in us. Not all organisms have this, but mammals and a lot of eukaryotes, we have exons and introns. Introns are spaces, so to speak, in between the coding regions that are going to become part of the actual protein sequence. And the exons are actually the regions that are in the final messenger RNA that end up making the protein. Now, as Dr. Wood alluded to earlier, a number of our proteins have the ability, or I guess I should say the cells have the ability to take certain exons and omit them or leave them in, so that you can get different versions of proteins, which is a really fascinating thing. In other words, when I entered grad school, our professors were astonished. They assumed that we would have somewhere around 100,000 or so genes in our genome. And I remember getting in the lectures there, and this is about 2004, 2005. I remember several of the professors getting up saying, "We don't understand this, but we only have about 20 to 25,000 genes. And we don't know why." But it was amazing. But as people started realizing the importance of alternative splicing, as Dr. Wood mentioned, that allows us to go from 20 to 25,000 genes to potentially many, many more gene products out of that process. So it's an interesting thing. But as we continue to zoom out, now we've got view of a full gene, and this happens to be one of my favorites. It's topoisomerase 2 alpha, so now you know why I picked chromosome 17. So TOPO2 alpha, here's the gene as a whole. The vertical bars are exons. The horizontal lines between those represent the introns. And so we've got, a couple dozen exons here that all get spliced together to make the protein that we call topoisomerase 2 from that final gene transcript. But we've zoomed out to a level. Look how far we have to zoom out. This is 56,000 base pairs now we're looking at. By comparison, just so you understand, when you have the final topo protein product, the chain is about 1,500 amino acids, which would be in nucleotide terms about 4,500 nucleotides. All right, so about 5,000 nucleotides end up, and we're having to zoom way out just to see what kind of space that covers. As we take another step out, 2.2 million base pairs is where we're at now. And now not only can you see the TOPO2 gene, look, now it's just a little bitty dot. Look at all the other green bars. Right? There's all kinds of genes around it. There's retinoic acid receptor alpha, ERB2, etc. So we're starting to see other genes. A bunch of them, they haven't put the names on there, but there's a bunch of other genes out there. So that's at the 2.2. So remember, we started at 80 million. You ready for this? So we step out again, or not. I'll just walk over here. Okay. So if we step out again, now we're at the 83 million base pair level. So we've gone all the way out, and now you've got all these little green bars, and then you've got a lot of spaces in between them. And those spaces are what we're calling some of that non-coding DNA, meaning it doesn't code for a messenger RNA transcript that becomes a protein. But it's there, right? It's a whole lot there. So the question is, what's it doing? And if you're thinking from a design perspective, yes, why would you want to have all this extra space, right? And for a number of years, folks from a design perspective have said it's got to be doing something. And to be fair, I think even people from a non-design perspective have acknowledged this. So we're not the only ones. But according to our paradigm, it ought to be doing something, right? It's not just there for no reason. And so the question is there functional information, in these non-coding spaces? Let me take an analogy from the computer science realm of what we call encapsulation. And so here's a concept where if you have data that you want to transmit, say you go open up your phone or your computer or your iPad and you go to the browser and you type in Google, right? And you want to pull up Google's web, homepage. So there's an application that you open up, you type that in, and that data has to go through a series of steps where there's a transport header added to it, a network added, a link header added, a link footer, all of these things. All you care about is this, but all the rest of that has to happen so that the information goes out to where it's supposed to get to on the internet, wherever that is, by the way. The internet just scares me because, I don't know, it's just there, right? Where is the internet? I don't know, it's there, right? So we send it out there to the internet, and it comes back to us as the data that we're interested in, right? What we're looking for. But all of this other information is needed in the process of that, of getting to that. I want to suggest to you that there's an analogous concept here in that while we have the gene that we're interested in, let's say, it may be bounded by other elements that are needed in the three-dimensional space of the genome to actually organize that information and arrange it. Does that make sense to everybody? You follow me then? So there are now what we know as insulator sequences. There are various types of regulatory sequences. There are promoters. And some of these sequences are very repetitive in nature, as Dr. Wood referred to earlier. And so let me suggest to you that if we combine all these components together, we may be able to start defining, and some people have started defining what various components in this arrangement are, so that we can understand when we look at the non-coding DNA, what are we looking at? Now, we owe a lot of thanks to the NIH and many other organizations that came together in these huge consortiums, not only to sequence the genome, but also to start studying function. And so the ENCODE project, which probably I should say instead of 2003 to present, I guess it's technically has concluded on some levels its work, but in essence still goes on. But this concept of the encyclopedia of DNA elements is basically where the researchers went back in and looked at the data and said, all right, what are all of these non-coding regions doing? What are their functions for them? Are there things going on? And of course, the paper that made the big splash was when they released a large batch of this data back in 2012. And this one happens to be the Nature paper associated with that. There were several papers, of course, that came out, including several Nature papers. But they said as they studied the various regions of DNA, they mapped regions of transcription, transcription factor association, chromatin structure, histone modification, and they suggested they could assign biochemical functions for 80% of the genome, which is really huge, right? That's a big deal. And it's hotly debated. So understand this, this was a big announcement, a huge issue, because 80%, when we used to consider that only 2% was needed for proteins, it was thought maybe there's about 10% of it that's functional, and the rest of the other 90%, was probably not really functional. So to say 80% is functional was a huge deal in its day, and still is. And even back in 2013, Dan Grauer, who's an evolutionist, said at a meeting, he said, look, if the human genome is devoid of junk DNA as applied by the ENCODE project, then a long, undirected evolutionary process cannot explain the human genome. If, on the other hand, organisms are designed, then all or as much as possible is expected to exhibit function. And I think that's a fair statement. And then he said, look, if ENCODE is right, then evolution is wrong. That's a really powerful statement, one that we need to at least stop and consider for a moment. Now, to Grauer's point, I don't know that we can fully say that all 80% of that genome is really functional unless you understand how function is being defined. And that's really where kind of the rub of this is. How do we define function? The classic definition was the central dogma, right? Function is the DNA codes for RNA, which codes for protein. But we're having to realize it's much more than that nowadays. So to understand the genome more holistically, we're going to have to understand that it's more than just about making proteins. It's also about the physical arrangement and organization of it in three-dimensional space. It's also about the regulation of it that has to go on in a cell type specific manner. Right? You've got 200 plus different cell types, depending on who you talk to, hundreds of different cell types in your body. How is it that those cells express the proteins that are needed to be expressed in a temporally regulated manner, in a spatiotemporal manner, etc.? So all of those things have to come together. So let's talk about some of the findings of what's been identified in terms of function in non-coding DNA. So again, non-coding DNA we're defining as those regions of DNA that don't necessarily code for proteins, even though much of this is expressed as RNA, and that's another thing that we're trying to wrap our minds around. Vast regions of the genome appear to be transcribed, not just the sections making messenger RNA. Other areas are expressed as well. Why is that, right? What are they making? What's going on? Is it haphazard? Is it accidental? It seems to be too much to be a random coincidence. It seems to be too pervasive to be accidental. So, we're talking about introns, repetitive elements, regions we might call pseudogenes, etc. And the question is, do they actually serve a function? A review back in 2012 suggested a number of possible functions for introns, including transcription, initiation, termination, genome organization. I thought this one was really cool, time delays. Some introns are ridiculously large, and there's some thought that that may actually be a timing mechanism in transcription that actually allows for the proper production of the products. And so it's very interesting to consider that some of the things that seem to be out of place in the genome may actually be there for a structural or functional purpose. So, even things like export, cytoplasmic localization, etc. You could do similar studies. This is a very well-known review by Shapiro and von Sternberg from 2005, and I recommend you look up this reference. I've just kind of screenshotted a small part of one table, which there's some massive tables in this paper. But they go through the literature, and this is back in 2005, so this is a dozen years old. But in the literature, they examine various things, line elements, sign elements, etc., and tried to find various roles for where these things are playing. And so, in part, these were examples of transcription, but you can find throughout the paper a number of other things of how these are playing roles. So some of these are promoters, some of these are playing other roles functionally. So I just want you to consider that some elements of DNA have already been defined, but it doesn't appear that one sign being this doesn't mean all signs are this. So it has to be studied in a very detailed manner. And that's one of the challenges of the ENCODE project, is now to go back and ask the question of, okay, these higher level studies, can we get down into the details on a lower level and actually verify and validate that data that it actually is functionally relevant? And that's the process that will be, I think, ongoing. An example from pseudogenes, just really briefly, the idea that pseudogenes would be a gene that maybe looks like it could be functional, but something is broken in it. It may look like another human gene. It may look like a gene in a different organism that maybe they have that's functional, but in us it doesn't work. And so the question is, why do we have pseudogenes? Where do they come from? Are they byproducts of mutation over time? Are they there for a reason? At least in some cases, pseudogenes serve to provide RNA transcripts that can actually participate in things like gene silencing. They may actually stabilize transcripts and allow them to last longer. And so you have different possibilities. So this was a discussion of that from 2011. So pseudogenes are another interesting one, and I bring them up specifically because this is one of the areas, and Dr. Wood's very familiar with this, I'm sure, that folks from a theistic evolutionary perspective bring up. Why do we have pseudogenes? Well, let's look at them. Let's ask the question, why are they there for a reason? And I think it's a fair question to ask, and I think it's a good question, a good research question. Something that I think goes along with this, and I just want to make a note, why is it that we care about other RNA expression? Some of the RNA that's produced is used to make what we call microRNAs. And so this is kind of the process of making a microRNA by first transcribing it and then processing it into various steps, a pri, then a pre, then finally a microRNA. And then eventually that microRNA can be used to actually destroy a transcript, for example, and allow for silencing of a particular expression. Now, we've taken advantage of this on a research front, which is really cool. You can design small sequences that you can actually put into an organism to silence specifically genes. And so if you're studying a particular organism and you want to get rid of expression of a particular protein, you can design a silencing transcript that you can get into that organism, and it will enable you to stop the production of your favorite protein or your least favorite protein, whichever it is. So one of the cool things, though, that we're starting to realize is as we study microRNAs, we realize these are all interconnected into a vast network. And so this is just one example from a cellular senescence network regulated by microRNAs. All of the red dots are proteins, the green dots are microRNAs, and you can see the complexity of this pathway. There's a lot of interconnections and interactions there. And so it seems to me that microRNAs are not there by accident. There's far too many interconnections here for this to be a haphazard or a random occurrence. There's a lot going on. And again, this is a very complex concept. So we have a lot of different potential functions for non-coding sequences: transcription, start, stop, timing delay, structural and architectural roles, transport, localization, and possibly many other functions that are still being discovered. And so I give you that very brief review just to say, think outside the box. Those of you going into research or in research, as you look at these non-coding regions of DNA, think about what could this be doing? What else could it be? What other roles could it play in this process? And consider that. It's also important to consider that a number of DNA sequences appear to be multifunctional. What do I mean by that? There are, for example, genes, and I'll show you an example of this in a minute, that are overlapping. Same sequence of DNA, two different start sites maybe, end up making two different things. That's a really interesting concept. That's like writing a sentence that means something when you start at the beginning, but if you start maybe in the middle of a word somewhere in the sentence and keep going from there, it means something else. That's a really complicated concept from a language and information standpoint. Same thing is true for this. A number of parts of our genome are transcribed bidirectionally, meaning one strand codes for one thing, the opposite strand codes for something else. How cool is that? Right? Because now we have a high constraint on the information. Think about how careful you have to be in your nucleotide placement, in the arrangement of your nucleotides, to code for a protein sequence on one strand and a different protein sequence on the opposite strand in the same region of DNA. So there are examples of that in our genome. There's also this idea of what some people have called duons, dual-use codons, with the idea being some codons are present to make, let's say, a protein, but also serve as a binding site for some other factors. And so the importance of those particular codons being those codons is not just to make the protein, but also in some kind of protein-DNA interaction. And so these are just kind of cool concepts I want you to think about when you look at the genome, to realize there's a lot of other things going on here. And I'm interested in what else are we going to find as we continue to study and ask these questions. So here's the example of overlapping protein sequences, a couple different gene examples here. Probably, my opinion, the best studied one would probably be this INK4A P19ARF concept, where you've got exons here, that get stitched together to make one version, and then you've got these other exons that get stitched together to make another version of the protein. So there's two different proteins being made out of the same region of DNA. This one's important because mutation in it, there's some association with cancer and basically cell cycle regulation, things like that, when we have disruption of this particular gene. All right, so those are all things in the genome that are going on. What about this idea of the epigenome, above the genome? And I put up here, this is the homepage of the Roadmap Epigenomics Project. I think this is a really interesting thing, and I think we understand still very little about the process of how it's all regulated, but depending on who you talk to, you'll get a little different answers on what are we talking about in terms of epigenetics. Some people define epigenetics as markers that are passed on heritably from one generation to another. On another level, though, there are epigenetic modifying regulating factors that aren't necessarily passed on in a heritable manner, but are present within cells and cell lineages. All right, so the nuances of how you define epigenetics is going to define how you actually look at it. But what are we talking about in practice? What we're talking about, generally speaking, are things like DNA methylation, so actually modifying nucleotides with methylation. That's actually part of this model here. These methyl groups added on here are kind of trying to represent that modification. And so this is something that can be added or removed by enzymes in our cells. and it has a functional consequence to the DNA. It actually influences how that DNA is used, whether or not the methyl groups are present. But then you also have this idea that DNA is not just present as a double helix, right? We've got DNA wrapped around histone proteins, making what we call the nucleosome. So in our nucleus, it's not just a double helix. It's a double helix that's basically wound around. It's almost like if we have a cord organization tool here where we're wrapping our cords up, right? Maybe some of you, if you take Christmas lights down after Christmas, you wrap them around a cord wrapper. If you have a long extension cord, you might wrap. That's kind of the idea of a nucleosome. Now, there's not a whole lot, we're talking about less than 200 base pairs wrapped around in a nucleosome. So you got to have a lot of nucleosomes strung together. So imagine if you had a really long extension cord that, okay, we wrapped it around that one, now we got it wrapped around another one, now we got it wrapped around another one, and on and on. What's controlling whether or not the DNA stays wrapped around that or kind of gets relaxed and released from that? Well, it's several factors, including modifications to the histones. So the histones make up the proteins in the core of that. They have tails, meaning ends on both ends of the protein that can stick out from that core, and those tails can be modified. We can have a methyl group, an acetyl group, phosphorylation, ubiquitylation. All these different modifications result in different things. So that's what they're trying to show you, these little tails here sticking out from those histone groups. And different modifications result in different consequences, meaning they either make the DNA more or less accessible. Why is that important? Well, this is controlling whether or not that region of the DNA is going to be used or not. Does that make sense? Now, why is that important in the life of a cell? If you just step back and think about cells for a minute, remember, we've got a bunch of different cell types through our body. You've got, your cells in your gut. You've got heart, lungs, brain, skin, bones, muscle. All these different cell types have to have different proteins to do the jobs that are needed in those cell types. So these modifications are helping the cell open access to or remove access to certain regions of the DNA so that that information can be used. All right, so it would be, by analogy, if we had, just think of it like a library for a minute. Do you want every book in the library open all the time? No, you put them on the shelf, right? You close the book, you put it on the shelf. You pull out a book as you need it, and you open it up. So by analogy, these epigenetic modifications are closing and shelving certain books and pulling other books off the shelf and opening them up. Does that make sense? And so and this is a dynamic process because it's happening in different cell types. It's happening in a time-dependent manner. It's happening in a cell type specific manner, and it's continually going on throughout our cells. So these modifications are being added or removed, and the books are either being shelved or pulled off the shelf in kind of an alternate manner, back and forth. So, does this have health impact? And the answer is, of course, yes. So there's a gene known as the MLL gene, which has H3K4 methyltransferase activity, meaning histone 3 lysine 4 methyltransferase. So it adds a methyl group to lysine 4 of histone number 3. And if you mutate that, you can get hypermethylation, a particular type of mutant. If you get hypermethylation, you actually get downregulation of something called tumor suppressor genes, meaning we shut those genes off, and those genes are responsible for helping prevent cancer. So guess what happens? These people develop cancer, right? If you have inhibition of tumor suppressor genes, you have now the ability to develop the cancer at a higher likelihood than normal. And so there are trials and things for drugs associated with that. So we know that histone modifications and epigenetic modifications are having an impact. This is one example. There are certainly many, many more. To the extent that you can actually find there are a number of biotech companies that specialize in producing products to allow you to study your favorite modification. Right? Say you're really interested in histone 3 lysine 4 methylation. There are groups that will sell you products that allow you to actually examine that modification. So it's You know when the biotech industry starts investing money in it, because they're trying to keep a beat on where are people going with their research, what are they interested in, and let's kind of follow the money, so to speak, in terms of that, so you know that they're interested. This brings me to another really interesting concept. Dr. Rod Carter mentioned this in the film, and so I have a little image of that here, but I want to expand on this idea a little bit, because it's a really important question. How are chromosomes organized in the nucleus? We said the other day, some of you may have been in here, each of us has about 6 feet worth of DNA in a normal body cell. 6 feet. It's a lot of DNA, compacted in a really tiny space. So we're talking about a rope, or a series of ropes, that are miles and miles long crammed into this room. I don't know, 100 miles long? I don't know what the analogy would be for the room of this size, but just imagine hundreds of miles of rope crammed in this room. What are the odds that that rope stays untangled? It doesn't get mixed up. What are the odds that a transcription factor can find where it needs to bind to produce the right protein from the genome? Right? You know, the RNA transcripts are produced in a very specific manner in order to make our protein. So what's regulating all of that? So a number of papers have come out over the last 10 or 12 years developing methods for examining how do we know certain regions of the genome interact with other regions of the genome. So there's this mapping technology, and this one happens to be Hi-C. So comprehensive mapping of long-range interaction reveals folding principles of the human genome. And what they did was really cool. It was very elegant from a methodology perspective. They said, all right, let's take DNA and let's cross-link it. And then we're going to cut that DNA with restriction enzymes. And then we're going to add a little biotin marker here, and we're going to loop these pieces of DNA together that have been cross-linked. And then we're going to pull down the biotin, shear up that DNA into fragments. And now notice what was originally 2 different pieces of DNA is now fragments connected together. And they get a DNA sequence of that region, and now they've got part of gene A and part of gene Y, and they're right now attached to each other. What does that tell them? Well, the inference is that now in 3-dimensional space, that means gene A and gene Y are really close to each other in the genome, in the nucleus. That makes sense? So in physical 3-dimensional space in the nucleus, those 2 things are close together. And they use these kind of heat maps to map that. Now you can do heat maps of a single chromosome to itself. You can do a chromosome to another chromosome. You can do all kinds of different analyses to figure out what parts are really tightly associated. But it's interesting, you get this kind of patchwork-like pattern. What does that mean, right? So people are scratching their head trying to figure out what does this mean in terms of 3-dimensional space and how things are organized. One of the things that they suggest in this study, and I think further studies have continued to pursue this, if you take, let's say, your chromosome, for example, you could imagine an equilibrium globule that's just all mixed up, where there's probably knots and tangles, or one where there's no knots or tangles, there's no tangling, and the evidence appears to support this latter model. And it appears to be very transferable and heritable. In other words, this happens reliably and repeatedly. So it's not just by random chance that the DNA ends up. It seems to be that it's ending up in the same place every time. And if you stop and think about it, it means that our genome is packaged in such a way that when it gets unpacked after cell division, everything ends up in kind of the same area. I'm not going to say it looks exactly the same, but it's ending up kind of in the same area. And so they said you almost have these chromosome territories where you've got regions out here kind of in the gray that are closed, but then you've got these regions in the middle that are opened up and accessible. And as you zoom in further on those areas, you find that you get reliable and repeatable kind of globules within that. So it's we're zooming way in when we get to that point of looking at that globule, but if you zoom way out up here to kind of the chromosome level, you can see that there are kind of territories in the nucleus. So it's really interesting. This is the concept of the 3-dimensional nucleus. How do we actually map out where these interactions are? Why am I talking about this? Well, one of the reasons I think this is really critical is I think part of this is going to help us understand why there is so much non-coding DNA. In other words, the sequences outside of the coding regions or even in introns may actually be really critical in helping make sure this organization comes together. And there's already some evidence for that. I want to make a prediction, and I think there's some evidence for this, and Dr. Wood, you can, I would love to get your feedback on this. I think that the order of genes along a chromosome matters. Maybe not in an absolute sense in some regard, but the order of genes along a chromosome I think does actually matter. And why I think that is because we're finding that as the genome gets organized into these different domains, there are various loops and things where certain sections of chromosomes are expressed together, right? And these looping domains of, let's say, 200,000 base pairs may have a couple genes in them that all get expressed together. And so we're seeing this phenomenon continue to occur in various ways. So does the order of those genes matter? There may be some areas where we can swap it up and it may not matter. It's going to be really hard, I think, to test that and really validate that. But I think at least conceptually, we can see why it might be the case. This study back in 2014, for example, this was kind of a preview part in a PNAS journal for a paper that was part of this issue, where they suggested, all right, we've got these interaction territories, and within those, there are these kind of looping domains, and some of them are nonspecific, and some of them are protein-mediated, where now we've got kind of our loop. And I put a note here, maybe this is kind of our data encapsulation, right? This is our packet of data that we've encapsulated, and we've looped it out there. And now we can control whether that loop is available or not. Meaning in a given cell type, we can determine, is that going to be accessible for transcription or not? We can turn it on and off. So now we've taken this huge chromosome and we've lumped out a 200,000 base pair fragment or so, and we've said this fragment is under control together. And then downstream there'll be another section that'll be under control together, et cetera, et cetera. And so, again, remember the chromatin wraps around these histone proteins, or the DNA wraps around histone proteins to form nucleosomes in the chromatin itself. And all of that has to be dynamically regulated. So we have an example here. So here's kind of our topological domains where all of this is overwound, so it's transcriptionally inactive. And I call that kind of, in data terms, it's kind of like data compression, right? This is our zipped file, so to speak. You ever use zip file? So this data is compressed, it's unavailable at the moment. Whereas this other data is underwound, decompressed, and now available for use. And this matters from a three-dimensional topology perspective, and this is my topo tie-in. We'll talk about topoisomerases in a minute. But the idea is, we have a certain winding to the double helix of DNA, and you can have overwinding and you can have underwinding, and we call those positive and negatively supercoiled. And our genome exists at various states of supercoiling depending upon if that DNA is available or not. And the way I teach my students in pharmacy school about it is I'll take a rope like this. I've got actually one much bigger than this that I'll loop around a doorknob or something, and I'll wrap it around itself to make a double strand. And then I'll have two students pull the ends apart, and you can see the compression of the double strand as you get positive supercoils ahead of that. That has to be very dynamically regulated. So this ties in with the work that I'm interested in because I study topoisomerases, which are involved in this process of regulating how wound up the DNA is, so to speak, how tightly coiled it is. And so if you separate two strands that are wound together in a double helix, you'll get compression, and that compression results in supercoiling. And supercoiling is a feature of our genome. It's part of what we have in our genome, and it comes with the two strands being wound around each other. It's interesting. Watson and Crick, in a paper after they noted the structure, they said, look, we know that this brings challenges. To think that the strands are wound around each other means that, how do you get them separated, right? They understood there was a topological challenge to that, but I love their quote because they said something to the effect of, we do not feel that this objection will be insuperable. So in other words, they said there's going to be a way for the cells to deal with this, we just haven't found it yet. Sure enough, in the 70s, the first topoisomerases were discovered, and so now we have a number of different types of topoisomerases. This is an example of a type 1, this is a type 2. And the type 1 enzymes can cut one strand of DNA, the type 2 can cut both strands of DNA. And we can facilitate things like relaxation, unknotting, decatenation. So only the type 2 enzyme can do these latter functions. Whereas the type 1 enzyme, which can only cut a single strand, can only allow for kind of torsional strain unwinding. So this is a really critical thing in our cells, that this be properly maintained. And so my focus in my research is on topoisomerase 2. It's a dimer, so here's kind of a look at a monomer, and then here it is in the dimeric form with one monomer kind of non-colored and the other colored. So it makes this really neat structure. It's got a number of different domains in the protein. And its role is simply this. It actually takes one double helix of DNA and creates a break. It passes another double helix of DNA through that break, and then it puts the DNA back together and releases it. So its job is to grab DNA, cut it, pass another piece of DNA through, and put the DNA back together that it cut. This might be parts of the same chromosome. This could be two separate chromosomes. Right? Whatever is needed, basically. So this is why your chromosomes don't get tangled up. If we don't have these enzymes, you're not alive, because your cells cannot divide and segregate their chromosomes appropriately. So it's a really critical function here. And one of the things I've been really passionate about lately is understanding the interplay between these enzymes and the opening and closing of various regions of the genome. This is one of the reasons why I'm interested in studying that. Something that's not very well understood about the protein is this kind of end region of it. What is it doing? It appears to be very different among different organisms and different versions. And the question is, what is it doing? And I think it's playing a key role in regulation. And the reason I study it is it's a cancer drug target. If you disrupt this function, you disrupt the ability of the cell to survive and divide, so you kill cells. So it's a very widely used cancer drug target. In bacteria, we can target this enzyme and kill bacterial cells. So there are a number of antibiotics that use this mechanism. So both in cancer chemotherapy and antibiotics, we can target topoisomerases and disrupt function. Now the problem is there's a lot of side effects. I have a 2-year-old niece that's about to begin another round of chemotherapy for her leukemia, and she's going to be on a drug called doxorubicin. Doxorubicin is a really powerful topo drug, but it brings with it some really nasty side effects, like cardiotoxicity. Right? That's not a good deal, especially for a little 2-year-old. Right? So how do we help little children like Lila get the treatment they need, but not have all the bad side effects? And I think the key in this is finding a way to target more specifically topoisomerase 2 alpha as opposed to beta. So we have two versions in our cells. And that's one of the things that I'm passionate about pursuing, is trying to understand that. But in the midst of all of that, I'm trying to understand the interplay here, because what it appears to be is that beta may be more involved in kind of the opening and closing of chromatin, and alpha is more involved in cell division. So which one, if I'm a cancer researcher, which one do I want to hit the most? I want to hit alpha, because it's the one that's allowing cell division, cell growth, versus beta, which is kind of the maintenance housekeeping version, which is going to be in your heart and other tissues that aren't growing and dividing. So we can hopefully reduce side effects if we can more selectively target alpha. And maybe I'll talk more about that tomorrow. I read this paper from Creation Research Society Quarterly a while back by Royal Truman, where he suggested that cells could serve as information processors. And what would it look like if we considered cells as that? And I encourage you to get a hold. There's 2 papers from 2 different issues. But in the first paper, he provides about 8 predictions for future discoveries of cells if we approach them from this perspective. He said, basically, these are things I would predict to see. I put 3 of them here. There's 5 more, and I'm happy to help you get a copy of that paper. I think actually one of these may be available for free through Creation Research Society Quarterly. But I thought it was a really cool perspective, because he said, look, if cells are acting a lot like information systems, computer systems, what would we expect to see? What predictions could we make? And how could that then influence the directions of our research and where we head with this? He said, for example, cells will be found to be more adaptable than suspected to situations not encountered before. Many more forms of complex regulation remain to be discovered. They discover multidimensional forms of data storage and retrieval not known for computers, etc. And so the interesting thought, now I took what he wrote out in paragraphs, I took in as one sentence from us. So understand there's a lot more context to the statements if you read this full paper. But just the thought that could we look at cells as information processors? Could we look at the DNA and the non-coding regions of DNA as part of that information processing system and ask, why would we have so many lines, so many signs, so many other repetitive elements? This idea that some of our DNA is potentially viral in origin or something like that, is that really the case, or does it just look that way, right? I think these are fair questions to ask. So just kind of as a way of closing, if we think about the updated central dogma, we got the basics, right? But there's a lot more to it now, because now we understand that proteins actually go and make all these metabolites and other macromolecules that we would call the metabolome. And then we've got RNA molecules influencing transcription. We've got RNA molecules influencing translation. We got other long-range interactions, proteins influencing transcription, proteins influencing translation. And then we find also that we have metabolites, and I know my arrows kind of got out of a line here, but all of this interplay, it's not as simple as we once thought, is what I'm trying to depict. There's a lot more going on, and that's not a bad thing. It's cool. But trying to wrap our minds around all these interactions is pretty crazy. So, all right, I'm going to stop there, and I open up to any thoughts, comments, questions, research ideas, and directions. Thank you for paying attention and staying awake.